Hello and welcome dear viewers to the interesting topic on dissolution. The topic will cover the dissolution method development for the products with low soluble drugs or the products which contain the poorly soluble drug substances. Mainly the video will cover how the dissolution parameters are to be selected and how the dissolution method is developed for the formulations like oral tablets, capsules, and other formulations which contain the low soluble or poorly soluble drug substances. See, generally the BCS class 2 and BCS class 4, these compounds have the low solubility, and BCS class 2 have high permeability while BCS class 4 has the low permeability. Then if you consider these low soluble drugs, then you will be knowing that the IVIVC is possible, or IVIVC can be made possible or can be studied or can be done for the formulations which contain the low soluble drugs. That's why the dissolutions become very much important for the formulations which contain low soluble drug substances. See, if the intestinal permeability is high for the drug molecules like belonging to BCS class 2, then IVIVC can be more predictable. While if the GI permeability or intestinal permeability is less understood, or if the intestinal permeability is low, then it becomes difficult for the drug formulations containing the drug substances belonging to BCS class 4. But still IVIVC can be made possible. So IVIVC is more predictive for the formulations which contain low soluble drug substances. Then the main problem for the formulations containing low soluble drug substances is that generally the dissolution is not achieved, and dissolution is not achieved, that means either the rate is very slow for the dissolution or the extent is not achievable. For the low soluble molecules, the complete release is difficult to achieve, like getting the dissolution above 85% or 100%. That is the extent of dissolution or drug release. Also the rate or release rate or dissolution rate is low for the formulations containing poorly soluble molecules. And why this rate is slow? Because these molecules dissolves very slowly. If the rate of dissolution is slow, then the dissolution time required for the formulation will be more, or the dissolution media volume will be much more than the mild conditions like 900 ml or 1000 ml. The dissolution method with long duration or with higher media volume than the normal may give rise to problems in the QC for commercial production. See, if you give the mild conditions for the dissolution test, and if you are achieving 80% dissolution after 2 hours, then consider the production plant where number of batches are manufactured, and at that site running a dissolution for 2 hours will be time consuming and will involve a cost. Then if you consider the media volume about 1000 ml, it will also give rise to problems in the QC lab. So for the commercial productions, there should be a robust, there should be a transferable and a discriminatory dissolution method. And as the IVIVC is possible for low soluble molecules, it becomes very much important to develop a discriminative method. Also, the dissolution method should be such that it get completed within a specific time. For example, 1 hour for IR formulations and 12 hours or around 20 hours based on the ER formulation. So this is the generalized requirement for dissolution. Dissolution should be robust, it should be transferable, it should not involve much time, and it should be discriminatory. So dissolution becomes the very critical. Then the parameters which affect the dissolution are the dose of API, saturation solubility of the API, and the parameters of the dissolution testing, like RPM of the apparatus like basket apparatus or paddle apparatus. These parameters affect the sink condition achievement for the formulation. So sink condition, you might be knowing that if the media volume dissolve 3 times the dose, then you can say that sink condition is achieved. I have made a separate video on sink condition, so you can go through that and have the in-depth understanding about sink and non-sink conditions. For low soluble drugs, achieving the extent and the rate of release is very crucial. And how this rate and extent can be achieved? So first is, to achieve the sink condition, we can increase the media volume and we can increase the saturation solubility. So these two are the main parameters where we can work to get the rate and the extent of the dissolution. The rate and extent of dissolution can be achieved by attaining the sink and by increasing the hydrodynamics. Hydrodynamics of the dissolution means you can increase the RPM of the paddle apparatus. For example, you are working on 50 RPM and you are not getting the extent or rate, then you can increase the RPM from 50 to 60 or from 50 to 75 based on the formulation characteristics. So now let's see what we can do to get the rate and extent. So first of all, we have to do the pH solubility studies to generate pH solubility profile of the drug substance, and then you check in at which pH the drug is showing higher solubility, and then check whether at that pH sink condition is achieved or not. So if the drug substance shows pH dependent solubility, then the pH media showing high solubility can be tried for dissolution testing. For example, you are working on 3 different pH like 0.1 normal HCl with 1.2 pH, 4.5 pH acetate buffer and 6.8 pH phosphate buffer. And if you see that in acidic media you are getting the good solubility, so you can directly select 0.1 normal HCl as a dissolution media, and then you can use the mild dissolution conditions like 900 ml volume and 50 RPM for paddle apparatus. If the media is not there in which sink is not achieved, then other ways are there to achieve the sink condition. The media in which the sink can be achieved can be used in the dissolution test if the pH is within the physiological range. We should not use the pH which is outside the physiological range, like 1.2 pH to 6.8 or 1 pH to 7.5 pH around. If the drug substance shows pH independent solubility and if it is insoluble across the pH, then sink condition can be achieved with the use of surfactants. Now, if you take a example of drug molecule which don't have any ionizable groups, and if it is not having any pKa, that time it will be insoluble across the pH range. And for such molecules, surfactants can be used or tried for the dissolution testing. So first way to achieve the sink is media pH to achieve sink condition. Then second is media volume. Now if you have the pH at which the drug is having more solubility, then you can go for the higher volume. Like if sink condition is not achieved in 500 ml, go for 600 ml or 900 ml or 1000 ml. So these are the generalized volumes for paddle and basket apparatus and can be done with 1 liter vessels. Then higher media volume can be used like 2000 ml with 2 liter vessels. Then you can go for USP3 apparatus where 250 ml media is achievable, or 250 ml media can be used in each tube, and there are 6 such tubes. So 250 ml each tube will give you the total media of 1500 ml. So second way to get the sink condition is increasing the media volume. And if you are using the USP4 apparatus, that is flow through apparatus, then here the media volume is flexible and the media volume up to 4 liters can be used. So this is another option. Then increase in the saturation solubility to achieve the sink condition is the another option, and this option work very well for many of the molecules. So use of surfactants involves the selection of surfactant and the optimization of its level. There are different surfactants available, and for the selection of surfactant, the surfactants are required to be tried, and based on that the surfactant is to be selected. So first is surfactant selection. Surfactants are of different types like anionic surfactant, cationic surfactant and non-ionic surfactants. I have made a video on the surfactants for dissolution testing. You can go through that video to have the complete understanding about the surfactants for dissolution testing. So anionic surfactant is sodium dodecyl sulfate, or it is also known as sodium lauryl sulfate, SDS or SLS. Cationic surfactant is CTAB, cetyl triethyl ammonium bromide. Then non-ionic surfactants. So polysorbate or twins, generally the example is 2 in 80 or 2 in 20. These are the non-ionic surfactants. So use the different surfactant in the similar concentration to select the best suited for the drug molecule. For example, take 0.25% of the each surfactant and run a dissolution test and see which surfactant is showing the great or better solubility and the dissolution rate. So select that surfactant, and then after selecting the best surfactant, then select the concentration or the surfactant level. So critical micelle concentration or CMC is very important for the selection of surfactant. Generally use the surfactant slightly above the CMC. The composition, pH and the ionic strength of the media affects the surfactant selection and its level or its concentration. Then use of the organic media or non-aqueous media. Generally, the non-aqueous media or the media containing the organic solvents like alcohols is not recommended or not uh, possible for the dissolution testing, as these are not the physiological type of media. Use of alcohol, isopropyl alcohol, these are not recommended but depends case by case basis. If you have tried all the possibilities for getting the dissolution rate and the extent and it is not at all possible, then you can do the studies by using the non-aqueous medias or organic medias, and these are required to be taken approved, or it is required to be approved from the regulatory authority. For example, we will see a case study. The dissolution testing should be with the mild dissolution conditions like 900 ml media volume and the basket apparatus with 100 rpm or paddle apparatus with 50 or 75 rpm. Similarly, the USP3 and USP4 apparatus can be used with mild conditions. Consider a higher formulation with BCS class 4 drug, that is insoluble drug, which is insoluble across the pH range. So now here the example is with a higher formulation containing the molecule which belong to BCS class 4 and is insoluble across the pH. So test the formulation with different surfactant. Then select the best suitable surfactant and then test the formulation with varying levels and select the level slightly above the CMC. You have to select the surfactant, then select the level of the surfactant. Perform the dissolution in 900 ml or less media volume with paddle speed of 50 rpm. Here I have considered paddle apparatus, as paddle apparatus is used for most of the formulations. That's why considered example of paddle apparatus. If you run the dissolution at 50 rpm with the selected level of surfactant in 900 ml media, and if the dissolution is achieved, then stop the dissolution. Otherwise, go for higher rpm like from 50 rpm to 60 rpm or 75 rpm. Higher rpm can be selected and justified if there is variation in the dissolution at lower rpm or if there is coning issue. Coning means in this the formulation releases the uh, granules, or say for example the disintegration happens and the heap is formed inside the dissolution vessel, and because of that heap the variability is there in the dissolution results. So to rule out those variable results, generally the hydrodynamics conditions are improved by increasing the rpm. So from 50 paddle rpm you can go to 60 rpm and then to 75 rpm and check whether the dissolution is getting improved or not, and like that we can develop a dissolution method for poorly soluble drug substances and the formulations containing the low soluble drug substances. See, dissolution is the performance test. Dissolution test is required for QC. It is also required for the BioWeber, and it is a routine quality control test. It is required for SUPAC. It is required for IVIVC, and nowadays the regulatory authorities are asking for the IVIVC. So dissolution is the key step or main step for the product development and product commercialization. Also, dissolution is must for site transfers, technology transfers, and change in the formulation. So have a good learning about the dissolution testing and watch the videos which are available for the dissolution method development and the dissolution technology. So keep watching the videos on Pharma Learning in Depth channel, and always remember that dissolution related questions are asked in the interviews. Thanks for watching the video. Till then, take care and bye bye.